Welcome to MOOC course on Introduction to Proteogenomics. Welcome to the hands-on session of WebGestalt. In today's session, Dr. Bing Jiang will teach you about how the results and job summary can give you useful information. He will also show you how the enrichment analysis can be visualized in different forms. He will discuss about different types of network based methods like direct neighbor based approach, module based approach or diffusion based approach. So, let us welcome Dr. Bing Jiang for today's hands-on session on WebGestalt.
Goals name summary. So this is first, this is not enrichment analysis. Don't use this to report as your enrichment analysis. This is just simply a classification of the genes you submitted based on some pre-selected biological process, cellular component and molecular function categories. Give you an idea of how many, for example, how many genes are related to biological regulation and metabolic process or response to stimulus. These are the high level categories, yeah. Well, again, that's I think you are a very good representative of the users. We got this request from a lot of users. So yeah, that's on our to-do list to add to, yeah, we will do that in the 2019 release. And it's just basically similar to the pie chart you typically see, to classify genes. And then the important part is this enrichment results. Let's don't do the redundancy reduction yet. And then the default view is a bar chart. So basically this shows you the enriched categories. Because we choose top 10 options, that's why we get the 10 categories here, right? And on the y-axis, you have the enrichment ratio from the Fisher's analysis. And then you can this one can be downloaded if you right click, and then you can download this as PNG or SVG for your presentation or for even for publication. I think the quality is good enough. Or you want to maybe visualize in another way, you can visualize all your results in a volcano plot like this, and then you see these are the gold terms highlighted the top 10 categories. You also have the option to change it. You can change the label from the gene set name, like the gene ontology, which you don't understand what it is, to something is more descriptive, like the description of the gene ontology terms. If you don't have this volcano plot, then you are still using the old version probably. So you have to go to the website, and the old website is like this, and you have to use the WebGestalt 2019 beta version, or you just put the 2019 in your URL. Yeah, next, this is a completely new feature. And the programmer also did a very nice job, as you can see, because these labels are crowded. And sometimes you won't be able to see, right? But the good thing is you can actually use your mouse to move this around, to move this to the right place that you want it to be. And of course, now it's difficult to see where you can draw the link, and then basically you can rearrange them in the way that it can be used for publication. Click on the draw link. Yeah, and the axis is the log2 of enrichment ratio. So basically this is you have expected the number of genes, right, if you do random sampling. And then if you have an enrichment, what is the enrichment ratio? This is this. And then the y-axis is FDR in the log scale. But after you move this around, you can see now you basically have a very nice view of everything. You can keep the link or you can remove the link to get a clean view. And then you can download this plot that is very, yeah, minus log FDR. Yeah, yeah. So the smaller will end up with the higher, yeah. So this is the DAG volcano view, and then you can also do the DAG view because these genes are organized in directed acyclic graph in gene ontology. You can actually see this as well, but this is available in the old version as well. And from any of this, and sometimes if you end up with too many categories, some of these are quite redundant, as you can see in Go, right? It has this hierarchical structure, I mean, the parent term and the child term. So you want to simplify this, and for example, you can do the affinity propagation-based simplification. Let's look at this. Let's do a bar chart. You originally have 10 significant terms. But if you do the affinity propagation, you end up with 3. That means this will group the 10 terms into basically 3 clusters and only pick 1 from each as a representative. That will simplify your interpretation. And another algorithm we implemented is the weighted set cover. This ends up with 4. Either of this is useful for you to simplify your, for 10, I mean, it's not a problem at all, but you can imagine when you have 200, and this would be a very useful feature to look at. And now we have the overview of the results, and you can download and save. But of course, you want to understand why this is enriched, right? You want to look at the detailed result. You can click on any of this bar that will show you the detailed results for that bar at the bottom part of this. For example, this shows the apoptotic process. And then you can click on this. It's linked to the database, Amigo. So this will give you a description of what is a apoptotic process. And then here you have the FDR result, the p-value before the adjustment, and then you have the gene set size, you have the expected value, and the overlapped number of genes, and the enrichment ratio. So basically, the enrichment ratio is the overlap divided by the expected. And then you have also very easily understandable Venn diagram to help you to understand this result. And then all the genes in overlapping genes here are in this table. You can browse all the genes, and you can also sort them in different ways. So yeah, and then let's say you did this analysis and you want to share with your colleagues, or if you want to share with your supervisor, he just doesn't have time to run this analysis. So, and then you can click on the result download. So this will save the result as a zip file. If you open this zip file, it will include the HTML file and other files. And if you click on that HTML file, that will basically reproduce this exact result. So it also provides a very easy way to share your results with others. So yeah, it's very simple like this. But you can explore this in many different ways. And you can go back and then change the, let's say we did the analysis against the biological process, right? Now we want to do pathway. Let's say we do change the functional database to pathway type, and then we use a wiki pathway for the analysis. And maybe we change the cutoff from the top 10 to FDR 0.05. And then you can submit. So basically this will compare your gene list with all the wiki pathways. Let's see what we get. I'm using wiki pathway as a demo. Yeah, you can choose Reactome if you want, or CAG. But I'm using wiki pathway. Yeah. FDR, yeah, I think you can do 0.05, for example. Yeah, it's up. Yes, yeah, but usually, I mean, you can do up to some people use 25%, 0.25. That's the largest I think you can go. Yeah, but I think usually you go with 0.05 or 0.01. This is the result I got. So. Basically, it's very similar to the gene ontology analysis, but of course, we don't have the DAG because this is not gene ontology, so there is that directed acyclic graph structure anymore. But you still have the volcanoprot, and then you can still put the description here. And now you can see, because we have a lot of enriched categories, so it's more useful to apply the affinity propagation. For example, you have a lot, right, and it's difficult to go through, and then use affinity propagation, of which it will give you a reduced set that can represent, basically highlights the representative ones. And yeah, that will make your volcanoprot easier to look at as well. And another difference between the, this analysis and the gene ontology analysis is for gene ontology, they just put genes together, but without defining the relationship between genes within that gene set, right? But here, we can have the pathway map. Yeah, this is the result. So, but if you click on one of these pathways, and then if you click on the pathway ID, it will also show you the overlapping genes here. There are 6 genes here, right? And then you have the pathway map, and the genes will be highlighted in the map. I don't know why only one is highlighted here. Let's try another one. One second. Yeah, so the highlighted ones are like, because sometimes one node might have represent a complex or something like that. There might be multiple genes in one node. But this provides a very easy way that you have not only have the pathway identified, but also you know where your genes are located on the pathway. And this highlighting function is available in all of the pathway analysis, like CAG, Reactome, or wiki pathway. So that is the first example, and I think you can change the parameters in different ways. For example, we only look at the gene ontology, biological process, and the wiki pathway, and using a specific cutoff, but you are free to change those and do your own analysis. But in the next example, I want to show you how you can do the gene set enrichment analysis in WebGestalt. And specifically what we have here is a pre-rank based analysis. We don't do the differential analysis in the system. Rather, you choose your statistical methods and you do your differential analysis, and after that you get a statistical p-value that will allow you to rank all the genes. In this case, for the ORA, as I said, let's say if ORA, the input is very simple. It's just a list of genes. So that is basically you do a differential analysis or correlation analysis, and you identify 100, 200 genes, and then you copy paste here, or you put that in a text file and upload here. But if you want to do GSEA, let's click on this GSEA sample run. And what you need to have is something like this. You have every gene has a value associated with that gene. And in this case, for example, this could be a t statistic or minus log p-value, something like that. So that will allow you to rank the genes. But notice this, in this case, you don't set any cutoff. If you do a proteomic study, and then you identified 5,000 proteins, and then you have a t statistic for all the 5,000, you need to put every single protein in this, but in this format, one gene or one protein and a value. And then the system will sort the genes and identify the location of each gene for a specific gene set where the genes are located in the ranked list. So yeah, because of the time, let's just do the GSEA sample run. I think that will be easier than getting the file and upload. But when you prepare your file, it's basically same as this, just a file, text file with 2 columns. Remember, for ORA, it's just one column or just a gene list. GSEA always you have to have a gene and a value from your statistical test. For example, you can calculate the correlation of the genes to drug sensitivity, and then for each of those you have a Pearson's correlation, and then you can use that to rank all the genes. So this is, if you succeed in getting the result, I mean, the top part will be very similar to the ORA analysis. Basically, you still get the bar chart or volcano plot, or if you do the GO, you get the DAG of your enriched terms. The major difference is here, and rather than the Venn diagram that shows you the overlapping genes between your uploaded gene list and the genes enriched gene set. For example, let's see if we are interested in this focal adhesion. We click on this, and then it's let's say this is a ranked list. This is the data this is your input. So basically now, because every gene came with a value, then the system was able to rank all the genes from the largest value to the lowest value, right? Here you can see this is ranked list metric. This is the number 1 gene, this is the last gene, basically from the positive values to the negative values. And now for the focal adhesion genes, these bars basically represent where are the focal adhesion genes located on this ranked list. As we can see, there is a tendency for these genes to be located on this part rather than this part. Basically, that means there are a lot more up-regulated focal adhesion genes than there are very few down-regulated focal adhesion genes. That's why it's enriched at this part. Also, it has this scoring plot, enrichment score plot, and this part is the leading edge, meaning these are the genes that give you the enrichment signal, and these are the genes that are listed in the table at the bottom. There are a total of 53 genes out of the 128 genes that are in the leading edge that is giving you the enrichment signal. So, so basically, if you think about the Venn diagram and this, you can probably can help you better understand the difference between these 2 approaches. One approach, you have a cutoff, and then it becomes 2 sets, and then you do the overlap, and then you use a Fisher's test or hypergeometric test to check the enrichment of the overlap. But here you are not setting a cutoff, then you get all the values for all the genes, and then you are testing their enrichment in the ranked list. So I think this is again, I mean, you can also, at the top, there is a download button, and then you can also save and download and save and share your results. So finally, let's take a look at the NTA sample run. So the NTA sample run, the input is also very simple. It's just, again, a list of genes you are put, let's say you have a list of differentially expressed genes or some genes you can just put here. The idea is to say, I mean, if I identify, let's say we do an association study, we identify 300 genes that are potentially interesting, which one should I test first, right? Do an experiment first. And this will help you to answer that question. So there are two options. One is to do network expansion, but the other is network retrieval and prioritization. This prioritization is when you have a lot of genes, you want to pick the top, and then you do this. But this can also be used. Let's say, I can show you another example later. Let's say you have one gene, but you don't know the function of this gene. Then you can just type the name of the gene here, and then you can do the same analysis. That will help you to retrieve the neighborhood of that gene, and then do co-enrichment analysis to help you to predict the function for that gene. But let's look at this first. Yeah, I think the analysis probably will take very long because there are a lot of people submitting job at the same time. But this is the result. And if you do the analysis later, you succeeded in getting the result. It's like this, and at the top you have the job summary, and here you have the subnetwork. So yeah, if we go back to here, we see, well, basically. This is our input gene list, right? And the network, we chose the PPI BioGrid. So this is a protein-protein interaction database. So basically, we are mapping these genes to a protein-protein interaction network. And then first, let's see here. First, we retrieved all the genes that are included in the network, and then you get a subnetwork like this. And then you notice that in the top 10 genes, because of your parameter is, and you want to highlight the 10 genes, top 10 genes, right? Then these are the 10 genes that are highlighted, and then you can zoom in, and it will tell you, for example, the FN1 and ELN, collagen A1, and TGF beta 1. I mean, these genes are the top genes based on the network diffusion analysis. That means these are probably the hub genes, or have more connections to the other genes in this network than the genes that was not selected as for highlighting. On the right side, so basically this is an enrichment gene ontology analysis for the network, and it will tell you what are the enriched functions for these genes, and you can click on this. For example, this is response to wounding, and then those genes will be highlighted. So basically, it provides you a way to retrieve the network and also understand what are the major functions associated with the network, and then you can also visualize which genes in my network has that function. And at the bottom, you have all the genes you submitted, and their rank. Like here, you can get all the rank, like FN1 is number 1, and then THBS1 is number 2. But basically, you can have your complete list here. Yeah, here, this is a gene ontology enrichment analysis result for the network. You have, so basically the typical gene ontology overrepresentation analysis report result, similar to the ORA analysis, but focusing on the genes in the network and using the gene ontology biological process for evaluation. Yeah. So these are the three major functions for the WebGestalt. So again, the ORA and the NTA, the input are just gene lists. It's very easy. You can just copy and paste your list to here. But make sure you understand the parameter setting and get the right parameters, and then the result is very simple to understand, I think. You can download the figures for very easily from the interface.
I hope today's session was useful to you, where you got an idea about exploration of WebGestalt result. So, we learned that WebGestalt gives you data of gene enrichment, gene ontology, protein-protein interaction modules and pathway analysis. In pathway analysis, we learned that we can choose different functional database such as KEGG, Reactome and WikiPathway. The filtering of pathway analyzed data can be done with stringent criteria such as 0.05 fast discovery rate. I hope you appreciate that resources like WebGestalt are so precious, open access available to the community, where from your complex mass spectrometry or even NGS data set, you can try to now further get an idea about what is the best biological sense of your data, how to really get the best biology out of that, the complex mass spectra, how best now you can try to address the biological question which you originally wanted to start with. So, these resources are highly valuable. Of course, these short lectures and hands on sessions may not be able to provide you all the information, but more and more you make yourself familiar by using these tools, you will then appreciate that the same data set which you have obtained from these high throughput technologies can now give you some very novel insight and probably the right answer to the biological question which you wanted to address. In the next lecture, Dr. Bing Jiang will teach you about network analysis. Thank you.